Guys, this is Mr. Bennett. This is notes number 10, and we're going to focus on enzymes with this video. Now, this is kind of building on yesterday's video where we talked about proteins and nucleic acids. They were two categories of the macromolecules. And in tonight's video, we're just going to focus on proteins. There's one category of proteins known as the enzymes. That's what we're going to talk about. Now, in order to understand how an enzyme works, we need to talk about chemical reactions for a moment. So a little bit of a review. First of all, we need to understand that energy is the ability to do work. And as we talked about yesterday, activation energy, or E of A, that's the symbol, is the energy cost for doing the work of changing reactants into products. So in other words, if I have a reaction and it's say A plus B produces C. Okay? In order to get A and B together and have that reaction to occur, we have to pay the energy of activation. All reactions cost energy. In other words, all reactions have an E of A. Some of them are low, some of them are high, but all reactions cost energy. That said, for humans and for living organisms, if we had to pay full price for every reaction that we want to happen in our bodies, we would die. There's no way that we can consume enough energy to pay full price for every reaction. So there has to be a way that we're able to save some energy and pay for all those reactions we need. The way we do that is through an enzyme. Enzymes, again, are one category of protein. Since they are a protein, that means that their monomer, or the pieces that they are made out of, are amino acids. Okay? And we define enzymes as a biological catalyst. Catalyst we'll talk about in a little bit, but let me talk about this word biological. Biological means that it was made by a living thing. So something like, for instance, our cells, they make enzymes. So living things make enzymes. They're just a type of catalyst that's made by living things. The role or function of an enzyme is to lower the cost of the reaction. I just mentioned earlier on the previous slide that we can't pay full price for every reaction and expect to live. Okay, we have to find a way to reduce the cost. We do that by using enzymes. They lower the cost. Okay, they reduce the activation energy. The other thing I want to mention on this slide is that the way we name enzymes is kind of special. We name them after what they catalyze. Okay, so whatever their job or their function, whatever reaction they're reducing the cost of, okay, we use that as part of their name. And then the other thing we do is we give them a special ending. So the end of their name is special. It's ACE, A-S-E. That tells us it's an enzyme. So here we have polymer ACE, or polymerase. This is an enzyme that catalyzes the function of polymers. My trick for figuring out what an enzyme does is I always read the name backwards. In other words, ACE means enzyme, and then polymer. So it's an enzyme that makes polymers. Okay? Now, I said we'd talk about catalyst. What is a catalyst? Okay? A catalyst is anything that lowers the activation energy. So there are other substances out there that are not made by living things that have the ability to also lower the activation energy. However, enzymes are one category of these catalysts. Okay? You don't find them everywhere. You find them in living organisms. They're made by living organisms. Okay? So, if we look at this picture here, all right, we have a reaction that's occurring. Now, in both curves, whether it's blue or red, it's the same reaction. The way we know that is that we start with the same reactants and we make the same products. The main difference between the two curves, the blue and the red, is the height of this hill. Notice this is the starting energy, the energy of the reactants. Without a catalyst, we have to go all the way up here. So this would be our cost. Okay? Think of this as full price. So I'm paying full price for the reaction. Now if I run it with a catalyst, the hill, so to speak, is only this tall. Okay? So this is the discount price. In other words, by using a catalyst, I only have to pay this much. So this up here, that's what I saved. I can take that energy and I can use it to run this reaction again, or I can use it to run other reactions, but the point is I didn't have to pay full price. I have excess energy that I can use elsewhere. Now, for enzymes, the number one thing you need to understand, aside from the fact that they're a catalyst, is that it's all about their shape. Enzyme function is directly related to their shape. So if we look at this picture here, notice that this enzyme has a specific shape. It's got a little groove, it's got like a little bumpy thing sticking out, and these little shapes allow them to interact with the reactants. Okay, the reactants fit right into those shapes. So the three-dimensional shape is what lets them do their job. Okay. The other thing you need to know is that when we're talking about reactants interacting with an enzyme, they get a special name. They're called substrates. So the substrates, or the reactants, they plug into the enzyme, activation energy gets lowered, okay? They interact, and they form the product. When it's done, the product gets spit out, and the enzyme is ready to do its job again. So, just another picture, same deal. Here we have the enzyme, cruises over, interacts with the substrate. Substrate's a fancy name for reactant. So reactant plugs in, catalyst happens, meaning we lower the energy of activation, we get the product gets spit out, and the enzyme is actually reusable. It gets used again. So, let's talk about what's going on here. Well, we saw that a specific reactant was plugging into the enzyme each time, and that the shape of the reactant fits specifically into the shape of the enzyme. That means is that each enzyme type is specific to a reaction. So in other words, if we take this particular enzyme called DNA polymerase, this is an enzyme that catalyzes making the polymer DNA. Remember how the trick for reading it backwards. It's an enzyme that makes polymers of DNA. Okay, so it's all about the shape. All right, think of it like this. If you had a hammer, okay, so draw my really bad picture of a hammer. Okay, a hammer has a specific shape that allows you to put nails in, right? When it hits that nail in, that's the equivalent of running the reaction. Okay, enzymes, as they're doing that process, as they're helping that reaction happen, they do change their shape. That's what allows them to push the product out. Then they go back to their original shape so that they can interact with the reactants again. Second thing is that they are not used up by the reaction. They can be used over and over. So just like this hammer up here, I can hit this nail in, then I can use the hammer to hit this nail in, then I can move on and hit another nail in. They are reusable. Enzymes are the same way. You can use them over and over again. They are not consumed by the reaction. The final thing here is that each enzyme molecule can only catalyze one reaction at a time. In other words, just like the hammer can only put one nail in at a time. I cannot use this hammer to put all three nails in at a time. One enzyme, okay, can only be used to to catalyze one reaction at a time. What I'd like you to do right now is you're going to pause the video in a second, and you're going to draw a picture of an enzyme interacting with a substrate. On your notes, you have a little box. You're going to draw your picture there. Okay, so pause your video, draw it, then when you start it up again, I'll show you how to draw it. You can check your answer. So pause now. Did you pause it? Okay, I'm going to assume that you paused it. Okay, so I'm going to do my enzyme. I'm going to call it Pac-Manase because it looks like Pac-Man. All right, and remember, it's all about shape. So the substrate or reactant that you draw needs to fit into the shape of your enzyme. So this is my enzyme, this is my substrate or my reactants. And one thing I do want to point out is that this space in here, the place where the substrate plugs into the enzyme, is known as the active site. Why is it called the active site? Because that's where the reaction occurs. Okay, that's where the reactants plug in, that's where the reaction happens. So real quick, on your drawing, go ahead and label this the active site. Alright, story time. Set your pencils down, sit back, and listen. Alright, so I may or may not have said this in your period, but just so you know, I teach not only during the year, biology, but during the summer, I'm also the summer school teacher. So that means that if you don't pass my class, you're probably going to see me in summer school. Okay, summer school is kind of long. We cover a week and a day. It's very tiring. And a couple years ago, in order to reward myself for going in and doing summer school each day, I started stopping by Wendy's and I would get something called a Coffee Toffee Twisted Frosty. So every day I would finish up here at school and then I would drive over to Wendy's on my way home and I would get a Coffee Toffee Twisted Frosty. It was a pretty good dessert. I like coffee, I like ice cream, I like toffee, so it works, right? Well, they also had a really cool commercial. It was kind of funny or entertaining, so I'd sing the song, I'd go get my Coffee Toffee Twisted Frosty, I'd get home, I'd sing the song. My wife got irritated because I keep singing the song, but it was good stuff. And every day I would get one. And I don't remember exactly how much it cost, but let's say it cost $2.50 for my Coffee Toffee Twisted Frosty. So I'm paying $2.50 a day. So in a week, I'm paying like, what would that be, $12.50 for 5 days? One day I'm sitting there in my class, my students are working, kind of glance at my desk, looking for a pen, and I noticed that at the end of the year, the ASB gave teachers a free Griffin Gold Card. Now, I don't know if you know what a Griffin Gold Card is, but a Griffin Gold Card, if you purchase one, gives you discounts at different establishments. And while I'm looking at my Griffin Gold Card, I happen to notice that my Griffin Gold Card at Wendy's will give me a 10% discount. Okay? So immediately in my head I'm like, ooh, Coffee Toffee Twisted Frosty for less money, right? Amazing. So I think about it, I'm like, okay, $2.50 for a Coffee Toffee Twisted Frosty. If I show them my Griffin Gold Card, all of a sudden I get to subtract 10%. So quick math, turns out that 10% of $2.50 is 25 cents. So that means that my Coffee Toffee Twisted Frosty costs $2.25. Okay. What that also means is if I use this 10 times, I've saved enough money, enough quarters, to get essentially a free Coffee Toffee Twisted Frosty. So sweet deal, right? So I start using my Griffin Gold Card. Drive over there, cruise up, order my Coffee Toffee Twisted Frosty, tell them I have my Griffin Gold Card, roll up to the little window. They charge me $2.25. Okay. Now the important idea here is it's the same Coffee Toffee Twisted Frosty. It's not 10% smaller. Okay. They haven't done anything weird to it. It's not missing the coffee or missing the toffee. It's the exact same dessert that I was ordering before. It's the same Coffee Toffee Twisted Frosty. Only I only had to pay 90%. I saved 10%. Okay. So here's the question. How does this relate to enzymes? Okay. I'll tell you. First of all, the Griffin Gold Card is an analogy for an enzyme. Okay. In other words, Griffin Gold Card works like an enzyme. How does it work like an enzyme? Well, it reduces the cost, right? So by using my Griffin Gold Card, I have reduced the cost of my Coffee Toffee Twisted Frosty. My enzyme, if I use that on a reaction, it reduces the cost in terms of energy. Okay. Also, it gives me the same product. Okay. So even though I paid 10% less, I still got the same product. I got the same Coffee Toffee Twisted Frosty. Third thing is that it's reusable. If you think about it, an enzyme, as we said earlier, you can use it over and over and over again. Griffin Gold Cards are the same way. Every single day I could go back, show them my Griffin Gold Card, and get 10% off every single day. So it's reusable. Alright? So what I want you to do now, pause the video again in just a moment, and there's a little space there. In a complete sentence, I want you to describe how the Griffin Gold Card is like an enzyme. Okay? So pause it right now. Alright, I'm assuming that you did that. You've got your sentence, you know what's up. Maybe use this slide to help you. Now we're going to talk about concentration. Concentration is how much of the enzyme you have. Or another way of saying it is how many copies of the enzyme you have in a given area. In a cell, in an area of the cell. Okay, it's how many copies of the enzyme you have. So if we go back to that hammer analogy, it's how many hammers you have, right? So hammers hitting nails. So each enzyme can only work on one reaction at a time. Just like the hammer can only hit one nail at a time, an enzyme can only catalyze one reaction at a time. If all the enzymes you have are busy catalyzing reactions, we say that you're at saturation, okay? Meaning you're at your maximum rate. Think of it in terms of hammers. If I have 50 hammers, that means I can hit 50 nails at a time, right? Each hammer hits a nail, 50 nails, okay? If I add more nails, okay, in other words, with the enzymes, if I add more reactants, I will not increase the rate because all the enzymes are already busy catalyzing reactions. Okay, giving them more reactants to catalyze won't do anything because they're all already busy. In terms of the hammers, think of it as if all 50 hammers are busy hitting nails, putting more nails down isn't going to make it go faster. Okay. However, if I add more enzymes, that will increase the rate because these new enzymes will be able to catalyze reactions of their own. Think of it in terms of hammers. Okay, if I have 50 hammers hitting 50 nails and I want to hit more nails, I need to add more hammers. So if I add 10 more hammers, I can now hit 60 nails at a time. Okay, so adding more reactants won't do much if all your enzymes are busy, but if you add more enzymes, your rate will go up. The last big idea I want to talk about with regards to enzymes is sensitivity. Okay, enzymes are sensitive. Okay, if you change things too much, they become damaged. The special name for that is denatured. So an enzyme that is damaged, meaning it has stopped functioning or it's not as effective, okay, is known as denatured. Going back to the idea of shape, remember shape is how enzymes do their job. So if we change the shape, the enzyme stops working. So denaturing an enzyme is directly related to or is caused by changing the shape. There are two major ways that you can change the shape of an enzyme or denature it. That would be to change the temperature or to change the pH. So changes in temperature or pH, as we get away from the normal temperature or the normal pH, okay, so in other words, if we get too high or too low, the activity will slow, meaning the ability of the enzyme to do its job will slow down and eventually stop. If we think of an example of a fever, right, our body temperature is normally 98.6, but as it goes up, our enzymes will slow down. They start changing shape as they get warmer. They start slowing down. They're not able to work as well. If our temperature gets too high, our enzymes are damaged far too much to do their job, and they stop working altogether. That's why high fevers are dangerous. It's also why when you have a fever, you don't feel so good. Okay, one of the things that's going on when you have a fever is your enzymes aren't working as well, which means you're having to pay more energy for your reactions. So you feel tired, you feel sluggish, you don't feel good at all. Okay, now going back to this idea of 98.6, that's the optimal temperature for our enzymes. Our bodies are designed so that we run at 98.6, so all of our enzymes are designed to work best at that temperature. This is one of the reasons why I said first unit, we need to know this term, homeostasis. Our bodies regulate our body temperature. They keep our body temperature at 98.6. Why? Well, last unit we talked about several reasons why. Here's a new reason. If we keep our bodies at 98.6, that means that our enzymes will be at optimal temperature, which means that they'll be able to catalyze reactions the best, okay, at the fastest rate, or they'll have the highest activity. Let's graph this. We'll take a little picture of this, or take a little picture view of how this works. So if we were to think of rate of a reaction, and along the bottom we had temperature, right? At 98.6, right, our enzymes work their best, so they have their highest rate. As the temperature goes down, their rate goes down. Their ability to speed up reactions goes down. If the temperature goes up, the same thing happens. Okay, pH could work the same way, okay, except it wouldn't be temperature here. For instance, if you're in the stomach and the pH is 3, right, as it becomes more acidic, the enzymes will be denatured. They won't work as well. If it becomes more basic, or it gets closer to 7 and then into the basic range, same deal. Activity will slow or stop. Alright, so those are enzymes. Okay, that's how they work. We will have a lab on enzymes. Make sure that now that you're done with the video, your notes are complete and you fill out the online questions. Alright, and we'll talk about enzymes tomorrow.